Fantastic. Well, welcome everyone, and thank you very much for joining us for this taster lecture. So this is the first of 3 taster lectures that we're offering this week. So tomorrow there's a lecture on vaccines, and then there's a lecture on, I believe it's plastics and sustainability on Wednesday, and Kim can give more details a bit later. My name is Dr. Qasim Rafiq. I'm an associate professor in the Department of Biochemical Engineering at UCL, with a focus on cell and gene therapy. And without biasing your thoughts already, I think this is the best topic out of the 3, given how exciting it is. But obviously my colleagues may differ, and obviously given the current circumstances that we're experiencing globally with COVID-19 and the search for a vaccine, I'm sure my colleagues tomorrow will make a very important point about vaccine development. But I think in the long run, cell and gene therapies are the way forward.
Sorry to interrupt. Just a very quick question. Somebody's asked whether the audio is coming through all right. Can you do me a favour and just let me know if the audio is coming through fine, and if it's just one person who's having a problem with audio? Sorry to interrupt, Qasim.
Okay.
Thanks. Okay, the audio seems to be coming okay for most people. So if you're having problems with the audio, I'm afraid it's your particular device. I'd suggest logging out and coming back in again if it's not working, okay? So sorry to interrupt, Qasim. Please carry on.
No, it's not a problem. So I'd like to welcome you all, and thank you very much for joining us. And I think particularly given the circumstances we're now facing, I hope you're all keeping safe and well wherever you are based. So today's going to be a lecture, or say an introduction to cell and gene therapy. And what I wanted to do was kind of give an overview of the topic, but also give you some teaching content that we actually deliver to some of our undergraduate and postgraduate students. So you get a flavor for the area and the teaching style as well. Normally in these lectures, whether they're online or whether they are in person when we deliver them at the university, I always try and make it interactive. Obviously, because we're using the Zoom webinar for this specific activity, it won't be as interactive as usual. But I normally do like to ask lots of questions and create lots of interesting discussions. So let's begin with cell and gene therapies, and I'm going to focus more broadly actually on biochemical engineering. So I've mentioned I am an associate professor in the Department of Biochemical Engineering. So where does cell and gene therapies fall into that, and why even mention biochemical engineering? So I'm a biochemical engineer by training. So I actually graduated from this department in 2008. I then left and came back in 2017 as an academic, as an associate professor. And it's always interesting when you've left and you come back to teach something that you've learned 10, 15 years ago. You can see how the field has progressed, and also you can see why UCL is where it's at and why it's doing what it's doing in terms of fantastic work. But why biochemical engineering? So biochemical engineering fundamentally is about making a discovery a reality. It sounds very straightforward, but it's one of the most challenging things when you think about what we're facing at the moment. And let's use the current example of the COVID-19 and the search for a vaccine. So there's many different centers around the world, whether that be Moderna Therapeutics in the US, whether that be Oxford or Imperial Universities in the UK, who are looking at vaccine candidates. So they're discovering and developing a vaccine to see if a vaccine will work. And we're actually collaborating very closely with the Oxford team on the next part. How do we make it a reality? And I won't go into that because that will be discussed in tomorrow's lecture. But how do we make therapeutic discoveries an actual reality? So we're not just treating 1 patient or 10 patients or 500 patients. We can treat the entire patient population that needs to be treated. So we can produce 10,000, 100,000, 100 million doses if needed. And that's something, obviously, one of the big questions at the moment in the search for the vaccine. How can we make enough doses once we found a suitable candidate? So I think when you look at biochemical engineering, colleagues and the work we do spans all of the different therapeutic modalities. So traditionally it's been antibiotics and things like penicillin and so on and so forth. And when you think about penicillin manufacture, I like to touch on this in depth because penicillin, many of you, if I ask you, I normally ask this question to everyone, but obviously it's a bit trickier. I normally ask, when you hear the name Alexander Fleming, what do you normally associate it with? And everyone says, penicillin. He discovered penicillin. But no one has heard, or very few have heard of the name of Sir Ernst Chain and Sir Howard Florey, who were 2 individuals who shared the Nobel Prize with Alexander Fleming for the discovery of penicillin. And the reason why they shared the Nobel Prize is because although they didn't discover it, they had the fundamental role of getting it so that it could be beneficial to people. So actually manufacturing and developing a manufacturing process. So Sir Alexander Fleming had discovered penicillin, but couldn't produce anywhere near enough to treat a single patient. Whereas the work that Howard Florey and Sir Ernest Chain were involved in was actually trying to take that discovery and make it a reality so that people could actually benefit. And that for me is fundamentally what biochemical engineers do. And in the cell and gene therapy research and the work that we do, that's fundamentally what we are focused on. It's all well and good discovering something that treats one patient, but how do we really benefit people in the long run and benefit the patients that need it? And that applies to vaccines for AIDS, or in this case for coronavirus, as we currently see, antibodies for cancer therapy, stem cell therapies to reverse blindness, gene therapies, and so on and so forth, which we'll come on to. So then, we've discussed briefly antibiotics, antibodies, and cancer therapies and so on, but why cell and gene therapy manufacture? So I like to do this. So we're not just going to focus on the core content and the learning knowledge, but it's important to put these things into perspective. Why is it that we want to develop new cell and gene therapies? Why should the UK develop these therapies? Why is it an exciting area moving forward? And one of the key reasons is that it is developing a high value manufacturing activity. That's a new term that's come out in the last 10 or 15 years, this concept of high value manufacturing. So many countries, particularly in the Western world, can no longer compete for commodity-based manufacturing. But where they are trying to develop value and generate health and wealth for their respective countries is by focusing on things like high value manufacturing, where it's very expensive, but it requires expertise and knowledge that not everyone has. So the UK, and the UK government in particular, has identified cell and gene therapies and more broadly regenerative medicine as one of the high value manufacturing areas. So there's been a significant amount of UK national investment, and actually there's now a huge amount of global investment for both governments and companies who are investing into this space. So all of the major pharmaceutical companies are now heavily investing in developing a cell and gene therapy or regenerative medicine pipeline. But there's also significant development potential. So one of the key challenges with getting these therapies to market is that they lack the real manufacturing platforms. So there's been some phenomenal clinical work, and I'll show that in a few slides. But there's been a lack of real translation. How do we get this to the patient? How do we make this cost effective? How do we make this affordable for the patients? And that's really kind of where the key challenges are moving forward, and I'll touch on that a bit later as well. So this was actually, and again, I like to do this, I don't want to just give the students and read it. Let's put this into context. This was a report written by Sir David Cooksey back in 2006, which identified actually the UK is very poor at translation. And this applies to many countries around the world, where we come up with amazing discoveries, but we can't actually manufacture them, or we can't actually generate value from them in the long term. And so Sir David Cooksey, in his report, a review of UK health research funding, identified that actually there is a significant gap in translational research. And the idea is, how do we get these therapies into the healthcare pipeline? So basic research is what many people are focused on here, but actually how do we get this through to preclinical development? How do we undertake the clinical trials? And then fundamentally, how do we get this into the healthcare delivery? And he found that there was a gap here in developing the preclinical studies, and there was a gap here in this second gap in translation. And that for us is what we want to address with the work that we do, but also within the cell and gene therapy space. That's where the major opportunities lie. And this, I think, highlights the need for these types of therapies. So there's been a huge amount of excitement. So this is an article published in the BBC News on the 5th of September 2018, where the NHS approved that they would fund a particular therapy, which I'll come on to a bit later, called a CAR-T therapy, which offers a cure for cancer. For certain forms of cancer, it offers a complete cure. Now, this is unheard of. Many of these patients were suffering from, in this case, acute lymphoblastic leukemia, a type of blood cancer, had no other treatment option. They'd had chemotherapy, it failed. They'd had bone marrow transplant, they failed. Effectively, they were destined to die of their disease. But through this revolutionary, effectively, gene therapy, they were cured, and they are still living to this day, completely healthy and free of any remnants of the cancer returning. And that just shows the potential. But the challenge is that one treatment costs over $400,000. And I'm not sure how many of us would be able to afford $400,000. The reason why this was very important is because the NHS agreed to fund it. So the NHS said, actually, we will fund it for UK patients who suffer from this disease, who are eligible for this treatment, we will fund it. Now, the reason why the UK did this is for two reasons. A, the UK wants to be one of the leading players in the cell and gene therapy space, as do the US and places like Singapore and Germany. But also, it's because they knew actually there aren't that many patients that are eligible for this treatment. I think there's only probably about 10 or 15 in the UK that are eligible for this particular treatment because of the stringent criteria. And so therefore, although it's $400,000 at this stage, they know that they're not going to have to spend huge amounts of money in the long run because there's only 10 or 15 patients that would need it. But the idea is that the UK wants to set the pace and the standard, and we want to focus on bringing down those costs. So let's look at now what therapies are starting to emerge. Now, when I first I graduated actually from UCL in 2008, I realized I didn't want to go into do antibody production or antibiotic production or vaccine production. I was really excited by the field at the time, which was called regenerative medicine, and has now kind of morphed slowly into renaming itself as cell and gene therapies. And at the time, there was no industry. There was barely any products approved. It was a big risk on my part to kind of say, this is what I want to focus on. But I'm absolutely glad that I did, because you can now start to see, this is an image from the US FDA, the Food and Drug Administration, the US regulator, which shows you some of these therapies that are now being approved or have been approved to treat certain forms of disease involving cell and gene therapy products. This is the US. In Europe, which the UK is still part of as part of the European Medicines Agency, the EMA, they recently approved 2 CAR T-cell therapies, which I'll come on to a bit later. And then this is, as I mentioned earlier, the UK, the NHS made a deal with the company Novartis, who developed that CAR T therapy and made it available on the NHS for children who are suffering from this condition. But what is involved in the biotech drug development cycle? And as I showed in one of the previous slides, much of the research is let's discover new biological entities. But the focus really in cell and gene therapy is we've got lots of new biological entities which are being considered or being developed or being investigated. The challenge is, how do we really start to get it to the market? So let's take the COVID-19 vaccine as an example. Oxford are developing their vaccine candidate as we speak. The challenge is, however, they've just started their phase 1, but how do they develop and manufacture enough material to go into their phase 2, into their phase 3, and make sure the material is of appropriate quality? That can take quite a few years. But obviously, in this case, things may be expedited for the COVID-19 vaccine, but in most cases, you have to go by a strict process to ensure that the medicine is both efficacious, as in it does what it's meant to do, but it's also safe. So these are some of the questions that we need to start thinking about very early on. How much do we want? When do we need it by? Can we actually make it at scale? How long will it take to make, and how much will it actually cost? So as I mentioned, there's various things that we as biochemical engineers make. We make penicillin, we make, even things like biofuels and so on as well. But fundamentally, it's about how do we get to that scalable manufacturing process so that we can actually develop these therapies for patients that need them. And these are really, as I mentioned, and the focus of this lecture is focused on these 2 novel and emerging therapeutic modalities. The idea of cell therapies, where we're actually using a whole cell as a form of a therapy. So we actually give patients cells to treat them of their condition. So for example, that may be stem cells and other cell types, and also gene therapies. So the idea that actually we can potentially cure certain conditions by either inserting or improving or modifying a damaged gene that might be in the patient. And this is particularly important for things like rare diseases. So at the moment, for most rare diseases, there's no cure. There may be some treatment option which helps them, but doesn't cure them of their condition. Gene therapy has the potential and has proven in some cases to cure certain conditions. And again, we'll come on to that in a bit. So I like to give this to the students. We don't read all the text, and I don't expect them to read all the text. They can read it after the lecture. But I think it's important to understand the definitions. And particularly in this cell and gene therapy space, there are a lot of different names used. So I've mentioned a few already. So I've mentioned cell and gene therapy. I've mentioned regenerative medicine. People will often refer to tissue engineering. There's, as you can see from the title of this slide, ATMPs, or advanced therapy medicinal products. Generally, they refer to the same thing. Sometimes I might be more specific. And here you can see specific definitions for each of these areas. So tissue engineered products, gene therapy medicinal products, cell therapies, gene modified cell therapies. And what I really want to focus on in this lecture are kind of gene modified cell therapies and gene therapies. Obviously, we have lectures that cover all of these areas and go into the regulations, the manufacturing, and the fundamental biology. But just obviously for the sake of this lecture and for brevity, I'll focus on gene therapy and gene modified cell therapies, because that's where there is the most excitement and the most opportunity to have a real benefit to patients and also to the global economy. So let's go into gene therapy. So again, I like to start by introductions. And gene therapy is very much the introduction of normal genes into cells in place of missing or defective ones in order to correct genetic disorders. So it's bypassing a faulty mutated gene. It could be inactivating or knocking out a mutated gene, or indeed in some cases, it could be introducing a new gene into the body. And normally it can take place via one of two ways, what we often tend to call either in vivo gene therapy or ex vivo gene therapy. I'll explain what they mean. In vivo generally tends to mean in the body. So in vivo gene therapy is where, for example, we'll come on to an example later, where we actually put the gene directly into the area where it's needed. So for example, into the back of the eye to cure certain forms of blindness. We actually use viruses to do that, which again, don't get too scared. I'll go into in just a second. So that's an example of in vivo gene therapy, where we actually do the modification in the body. So we actually insert the gene via a viral vector directly into the back of the eye. Then there's what we call ex vivo gene therapies, where the genetic modification takes place outside of the body. So in this case, for example, with CAR T cells, we take T cells from the patient. We actually remove blood from the patient to get the T cells, which are in the blood. We then isolate those T cells, the specific cell types we want, and we then modify those genes, modify those cells, those T cells, in the lab. So we introduce a chimeric antigen receptor, a particular gene, which will have the therapeutic benefit or effect. And then we put those genetically engineered cells back into the patient. So we've done the modification outside of the body, but we're then putting the modified cells back into the body. So that's what we call ex vivo gene therapy. But ultimately, a very simple understanding and definition of gene therapy is the introduction of nucleic acids into a cell for therapeutic effect and or benefit. So what is the therapeutic strategy? What do I mean by that? So when we say therapeutic strategy is, how are we going to treat the patient? How will we have the benefit and the effect? So normally it happens via one of two ways. We either identify a faulty gene and replace it with the corrected version, or we can potentially silence a gene in the case of overexpression. So where a gene is being overexpressed by the body, we can try and silence that gene so it's not expressed. But this can have complications. Obviously, the germline origins of genetic disorders mean that every cell in the body contains a faulty copy of the gene. So if someone has a particular disease, for example, cystic fibrosis, it means every cell in that body contains a faulty copy of that gene. So this is kind of some of the key areas that are emerging about how we can address these issues. So let me just go back. So what is very much a focus is identifying and evaluating the rationale for the gene therapy approach, particularly within rare diseases, and starting to identify which diseases we want to treat early with the gene therapy before the disease progression occurs. But the next question is, and this is kind of, I've alluded to it to some extent, but it's one of the major challenges from a manufacturing or therapeutic delivery perspective is, how do we introduce the corrected gene into a patient? So really there's two main ways. I'm going to summarize the slide, but effectively to deliver nucleic acid material into the body, there's two main ways. We need to package it into some sort of vector or delivery device. And there's two main types of vector we can use. We can use viral or non-viral. And really for the purposes of this kind of short session, I want to focus on the viral delivery. A, because it's the most predominant at the moment, the most well understood and well being used to manufacture therapies for patients at the moment. So it's very much a focus, but also it will tie in with a bit of tomorrow's lecture, because again, part of the vaccine development is manufacturing virus. In that case, the manufacturing virus for a different reason. Here we're using a manufacturing virus to deliver a gene of interest to the patient to correct some sort of underlying condition. So now let's take a step back. If we're using viruses, why are we using? So the question hopefully you should be asking, you may already know, but for those of you that don't know, and you know, hopefully wondering, why are we actually using viruses, you know, at all? You know, what sense and what benefit does it make to use viruses? I mean, we commonly associate viruses with very horrible things. Coronavirus at the moment, and not excluded. So why are we even thinking about viruses? Well, really it comes down to the viral life cycle. And we're effectively using the virus as a tool because we know what the virus does, and it can actually do the job we want it to do. So normally a virus would invade a host cell. So let's take the coronavirus as an example. It would invade the host cell. What a virus does, it's a very parasitic organism. So it will invade a host cell. It needs to have a host. It will invade a host cell. It will then introduce its own nucleic acid coding for certain components of the virus into that host cell. So it uses, so let's say it enters into one of my cells. It will use my cell to then create copies of its nucleic acid to then generate more virus. That will then induce the host cell. So the host cell, so let's say my cell, will now express and manufacture the viral proteins, which replicates the viral genome. And the package, it then packages a new genome with a new viral proteins, and it leaves the host cell. And then the virus that has been manufactured using my cells, and now, you know, more virus has been manufactured, and those virus particles will do the exact same thing. They will invade other host cells until eventually my own body and my own immune system will mount an immune response. So my T cells will kick in, my NK cells will kick in, hopefully, which is why COVID has been such a major issue, because in many instances, and again, I'm trying not to steal the thunder from tomorrow's lecture, but in many instances, the body's own immune system, if it's compromised, isn't able to mount a sufficient response, which is why there are certain populations that are particularly affected and targeted. So viruses are effectively capable of targeting specific cell types. Now we've got that knowledge, can we use viruses to actually deliver something good to the cells? So can we deliver new genes to the cells using viruses? And so, you know, a few years ago, scientists thought about this, and it works very well. So the idea now, so instead of using the viral nucleic acids, what we can do is we can induce or introduce the gene of interest, so the gene that will make people better, that nucleic acid material into the host cell using the virus. And we can use the virus, which will then manufacture that gene. It will induce the host cell. So let's say my cell now, let's say I've got a genetic disorder. We can use the virus to manufacture the gene of interest. And now actually the virus is producing something useful. Now, obviously what we have to do very carefully and very importantly is make sure we've removed any of the dangerous aspects of the virus. And that's where a huge amount of work has gone into before it even gets anywhere near the patient, is making sure that anything that can be disease causing, so any of the nucleic acid material that causes disease, is removed from the virus. But what we're effectively using is we're using the delivery vehicle of the virus. So let's take a very basic example. Let's say the virus itself is composed of two parts. It's composed of the outer shell, so the envelope and the capsids, as you can see here, but it's also contained of the viral genome. Now, the outer shell and the envelope and the capsid, the way I like to describe it to my students is that this is like the delivery vehicle. So it's like a car. So that's the outer shell. You've got the car, it's delivering something that it could be delivering anything. It could be delivering something dangerous, it could be delivering something beneficial. But ultimately that's decided by what the viral genome inside the delivery vehicle contains. So if we can remove the dangerous elements from the viral genome and insert the beneficial elements that will benefit the patient, so the new gene, we can engineer the virus in order to do that. And therefore, that allows us to make use of viruses for the treatment genetic disorders. Now, we can use non-viral methods. So I mentioned there's two ways of doing it. There's viral and non-viral. And non-viral methods, personally, I believe in the next 5 to 10 years will be the main way forward. And people are already looking at how can we move away from virus, partly not so much because of the safety issues, because I think a lot of the safety issues have been addressed, more just the cost. It's very expensive to produce virus. So they want to move away from virus and they want to move towards non-viral methods. So again, some very clever scientists have thought about different ways of how can we disrupt the cell so that we can introduce material into the cell. So how can we, for example, allow nucleic acid material, so the gene of interest, to get inside a cell if we're not using a virus? So people have looked at electroshocking the cell to create mini pores within the cell. That's called electroporation. And there's various other non-viral methods. And again, I'm not going to go into that in detail. We have a whole separate lecture where we go through and we deliver to our master's students focused on non-viral technologies and non-viral approaches. So as you can see here, it's important to understand the general structure of the virus as well in order to be able to make use of its abilities. And I think one of the other key points to mention is there are different types of viruses. So as you would have seen from the coronavirus images, the reason why it's called a coronavirus is corona effectively means crown, and it's got a crown shape in structure. So these are different virus structures that we can see. So we have bacteriophages, the influenza virus, which is similar to coronavirus, has a particular shape, adenoviruses and tobacco mosaic viruses, and so on. So there are different types of viruses, and we can use them for different purposes. They have their relative advantages and disadvantages. And again, I'm not going to go into that now. That's perhaps too much detail at this stage, but is the type of detail we'll go into in our actual undergraduate and postgraduate programs. So there are different types of viruses that we can use, and they have different structures and shapes which can impact. And the question is, does size matter? And yes, absolutely it does. So the size of the virus limits the size of the genome that can be loaded, or the gene of interest that can be loaded. So this affects the choice when developing gene therapies. And again, so if you're thinking about how can we develop new therapies, then you have to understand the fundamental virology, because that will then allow you to make the right sorts of decisions, or allow you to think, well, okay, we can make these modifications with these types of viruses, and this should work based on the size and the structure of the virus itself. So now let's look at, I mentioned briefly already about viruses as gene delivery vectors. So here's again some key, I like to put this into slides. So this is actually slides developed by one of my colleagues, but this is the type of structure that we develop and for our teaching material. So we like to kind of give key glossaries and key information that students have this information at hand. So for example, for glossary of key terminology for gene transfer, when we're talking about the method of DNA transfer, we normally come across two types. We have transfection and we have transduction. So I've talked about non-viral and viral. The way we would term that technically when we're talking about the gene delivery itself is we'd refer to transfection if we're using a non-viral method to deliver the DNA into the cell. So we then say the cell has been transfected, and that's the process of cell transfection. Whereas if we used a virus to deliver the material, or deliver the DNA, we then call that transduction. So we have transduction and we have transfection, and that relates to the method of DNA transfer. We also have what's known as the duration of expression. So we have stable expression and we have transient expression. Again, I won't go into too much detail for the sake of brevity and the time that we have, and also the level that we're looking at targeting this material. But in our undergraduate and postgraduate courses, we go into more detail. But there's two types of expression which relate to the duration. There's what we call stable expression and transient expression. So stable expression is whereby the gene of interest is now fully embedded into the genome. So any daughter cells that are now coming in, future generations of cells that the body produces, they will all have that gene, because that gene is now fully embedded into the DNA of the cell. So anytime a daughter cell is produced, the gene of interest will also be produced. And that's what we call stable expression. We also have what we call transient expression. So sometimes transient expression is the only way we can do things. So it won't be embedded into the DNA of the cell. So when the cell does divide, it will mean that eventually the gene will get lost, because there'll be new daughter cells that will be coming through, and they won't have the gene of interest because it's not stably expressed. However, it's not always a major issue, because in many instances, particularly for mature cell types, they don't divide that often. So actually, it's not a major issue, but where possible, we try and look for stable expression. And then finally, there's the different types of viral vector. So we've mentioned, obviously, different viruses, but amongst the different viruses, we can classify the viruses into two types of vectors, what we call an integrating vector and what we call a non-integrating vector. So an integrating vector actually integrates the foreign DNA into the host cell chromosome. So we would use, now hopefully if you're following, we would use an integrating vector to achieve stable expression because that integrates the foreign DNA into the host cell chromosome. Whereas with a non-integrating vector, such as adeno-associated virus or adenovirus, in this case, integrating is an example of that is lentivirus. But for non-integrating vectors, where it doesn't integrate the foreign DNA into the host cell, but it just leaves it with it leaves it in the cytoplasm of the cell effectively, so it doesn't embed into the DNA. We call that transient transfection, and that's achieved by using a non-integrating vector. So sometimes actually people would prefer to use non-integrating vectors because they think it might be a bit safer. You know, it's not going to be there for the rest of the person's life. It will be there for as long as it needs to be. And then if needed, they can maybe get another top up of the therapeutic so that it can remain being transiently expressed. So using viruses as vectors. So a lot of the focus is how do we recreate the viral structure but replace the viral genome with the gene of interest that we're interested in. And the technical term for that is what we call the transgene. So gene of interest, the technical term, or people do often actually in scientific communities do use gene of interest. The technical term is effectively the transgene. So how do we insert the transgene into our cell? We can understand, by understanding the viral structure, we can replace the viral genome and use the virus as a vector or delivery system to deliver the GOI payload, the gene of interest payload. So the question is how do we effectively encode the recombinant protein in the new virus particles? And the other key aspect is we have to ensure that the GOI payload is packaged into all of the new viral particles and not the viral genome itself. So going back to the safety issue, we have to make sure that the virus doesn't have any of its original disease-causing ability. We've removed that. We want to make sure we've removed that. But it does contain the genome and the gene of interest that we're particularly interested in expressing. And so for those of you who are familiar with recombinant protein production, it's actually not that dissimilar. So the next slide briefly touches on recombinant protein production. So let's look at the production of insulin. So with insulin, for example, we isolate it from a healthy human cell. We isolate the insulin gene from a healthy human cell. We will take a plasmid from a bacteria. So plasmid, as you can see at the top, is closed circular bacterial DNA fragments. So we can take a plasmid from a bacteria. We can take the insulin gene from a human cell, a healthy insulin gene from a human cell. We can then combine the insulin gene with the plasmid to develop recombinant plasmid, which now has our insulin gene. We can then use bacteria to manufacture and actually effectively act as cell factories. So they're producing all of our plasmids which contain our insulin. We grow that in culture, normally at large scale, and then we can extract this insulin gene of interest at the end. That's how we manufacture recombinant proteins, and using insulin as an example. And those of you who have done GCSE or A-levels probably would have covered something like this. It's not too dissimilar when we're producing viral vectors, except in this case, the recombinant proteins, we're not producing insulin genes, but we're producing viral proteins. So the process is very similar in the approach. And we don't normally use bacteria cells. We tend to use mammalian cells. So in this case, we would often use human embryonic kidney cells or something of that like. So an example of the plasmids required for lentiviral vector production. So we obviously require certain plasmids. And if we go back to I'm going to go back. Hopefully it will allow me to go back and it doesn't jar your screens too much. So I'll let it settle for a couple of seconds. Hopefully it's caught up and you're on the right slide. So as you can see, we've got different parts of the virus. So we've got the envelope, we've got the capsid, we've got the viral genome, and we've got the envelope proteins. So we want to make sure that when we're developing our viruses as vectors, it has all of those aspects. And so when we're developing and identifying which plasmids we need a plasmid to develop the envelope. We need a plasmid to develop the packaging, what we call the packaging plasmid. That's what brings everything together. It brings the whole virus together at the end. And we need the transfer plasmid, which contains our gene of interest. So we've got three things there. We've got the envelope plasmid, which is the genome or the DNA, or in this case the RNA, that's required to produce the envelope for the virus itself. We've got the packaging plasmid, which are the genes that are required to actually package the virus at the end and bring everything together. And we also have, importantly, our transfer plasmid, so the gene of interest that we actually want the virus to produce. We co-transfect everything together, so these three different plasmids, and we then co-transfect and put this into human embryonic kidney cells to produce our gene of interest loaded viral vector. So in a very kind of random, hopefully relatively simplistic, obviously there's a lot more detail, and we get into a lot more detail, but that's the high level concept, and that's what we kind of wanted to go through. So let's look at a couple of examples just to round this off. So Luxturna is a viral gene therapeutic to treat LCA, so Leber congenital amaurosis, which is a horrible eye condition. And effectively, people end up becoming blind as a result of a lack of this gene. So it's targeting the LCA2 gene, which impacts the retinal pigment epithelial cells. And what we're effectively doing is delivering this gene. Now, this has been transformational. The patients who have received this, who were previously blind, have now started to have their sight restored. That, for me, you can't put a price on. Ultimately, you have to. But for anyone who is blind, to have their sight restored is incredible. And that is the type of transformational effects that we want to have. And now this is a product which is now available on the market. And just to prove that, in February earlier this year, so just prior to the COVID outbreak, we had colleagues at Great Ormond Street Hospital at UCL actually deliver the first ever Spark therapeutic gene therapy. So for this particular gene therapy, the first ever in the UK was delivered at UCL, at Great Ormond Street Hospital. And I met with some of the people involved, I think about 2 hours after they had transfused this into the patient. So some phenomenal work that they had done. And it just shows the impact that it can have. And hopefully, we expect that the individual who received that particular therapy will benefit from receiving that gene therapy. So I like to show that video for one reason, because it shows the impact of the work that we're doing. So that's what's known as a CAR-T therapy. And that's exactly what we're manufacturing at UCL, and it's what we're researching, it's what we teach our students. This is the latest cutting edge, therapies that are, was recently approved. And that's the therapy that costs $400,000. And the work we're doing at UCL, which we teach to our students, both in the undergraduate and postgraduate programs and in the research programs, is how can we reduce the cost of that so we can make sure thousands of children can benefit from this particular therapy. And the key thing about that was there was no other treatment option left for Emily. She would have died of her condition had she not received that particular therapy, and probably within a few weeks. And that was now, I think she's had her 10-year anniversary, I believe it is, which is incredible when you think the fact that she had weeks to live. So this was, when I showed the video, I like to show the second slide. So Emily, once she recovered, had the opportunity to then meet the president of the US, Barack Obama. And when you get an insight into her character, because when the president asked her, is there anything I can do for you? She wasn't allowed to tell anyone where she was going for security reasons. And she said, yeah, there is one thing you can do for me. My school don't know I'm here today because obviously I wasn't allowed to tell anyone. Could you write me a note so that I can give that to my school so that they know I was with you and I wasn't just missing class? So that's the note there from President Barack Obama to say, please excuse Emily from school. She was with me, which I think is always a nice touch as well. But that's the focus of the work that we're doing. So I'm going to start to wrap up very quickly so we've got time for questions, and I'll be more than happy to take questions afterwards via email or through Kim and our colleagues within the department. So why UCL for cell and gene therapy? So UCL is running more clinical trials in advanced therapies, so ATMPs and cell and gene therapies, than every other UK university combined. It's also running more clinical trials, the way I like to frame it, it's running more clinical trials than Italy as a country or the Netherlands. So it just shows you the amount of work clinically that's happening at UCL and the work that we're now engaging with our clinical colleagues and looking at how we can get these therapies to patients that need them. So here's just kind of an overview of some of UCL's capacity and capability to deliver these therapies to patients. So huge amount of investment, huge amount of activity and research. FIH stands for First in Human, so First in Human Gene Therapies for Rare Diseases. That we can now add a new one to that, which was the Spark Therapeutics Luxturna therapy, which I haven't added on to here. But there's a whole range of, you can see First in Human. But I think importantly at the bottom, all of these companies aren't companies that we collaborate with, although we do. These were companies that spun out of UCL. So companies that were developed by UCL academics or clinicians or students. And there's one here, Puridify, which is actually a spin out from our department as well. So for some of our former PhD students with one of our academics spun out this company, which is now being acquired by GE Healthcare. So I want to start to wrap up. Obviously, I mentioned about personalized CAR T therapies. There's a lot of news and BBC. There was a fantastic documentary, if you haven't seen it, on BBC Two called War in the Blood, which shows the patient journey of patients who received CAR T therapies as well. And then just finally, just to mention the research activity. So all of the teaching that we deliver is funded, or sorry, it's not funded, is premised on the phenomenal research activity that we have and the cutting edge research. So here's just one example. This is a £10 million activity provided to us by the UK government here at UCL to look at how we can manufacture personalized therapies, particularly cell and gene therapies. And to do that, we work with 35 different companies, some famous companies, GSK, Fujifilm. They don't only make digital cameras, by the way, they make therapeutics. So Fujifilm and so on and so forth, AstraZeneca as well, some of you may have heard of. The final couple of slides, I just want to briefly touch on the degree programs offered by UCL Biochemical Engineering. I realize we have a mixed audience. So we offer 3 undergraduate programs, a BEng and MEng in Biochemical Engineering, a bachelor's or BSc with a science and engineering component in bioprocessing and new medicines, and the same bachelor's, but this time with a business and management component, also within bioprocessing and new medicines. And for our postgraduate taught degrees, we have an MSc in Biochemical Engineering, which is kind of advanced biochemical engineering. We have a new master's program, which I'm leading on, and it's starting this year. I'll be happy to take questions on that afterwards, or certainly after this lecture as well. And you can find out more information on our website. We have an MRes in synthetic biology, and we also have an MSc in biointegrated design. So with that, I'd like to conclude and thank you all for joining us. I'm happy to take any questions, and apologies for overrunning slightly, Kim, there. And I'm just flicking through the questions now. There's some fantastic, really good technical and scientific questions. I'm really impressed, really genuinely impressed. It's fantastic to see.
And there's a cluster on ethics as well, which I thought seemed to, there seemed to be sort of similar theme coming through on that.
So I'll touch on that very briefly. The ethics are fundamental in everything that we do. And in many times, students don't get taught the ethics, or they're not kind of exposed to the ethics. And for us, in our new module in particular, we've got a whole module which is called Commercialization, Regulation, and Ethics of Stem Cell and Gene Therapies. And to make sure we're teaching the latest breaking content, the individuals who are delivering that are ethicists who are involved in the field, but also experienced lawyers who are involved in generating the ethics and the regulations around that. Because we feel that is fundamental to everything that we do. For public acceptance, we want to make sure the work that we're doing is ethical, it's safe, but also it has the desired therapeutic effect.
Yes. Well, we've got so many questions here. It's difficult because obviously there's some clustered together for that, and then there's some quite focused questions. I think perhaps if I start off something that's very, very topical, and ask about how COVID antibodies were extracted from human blood plasma. Is that something which is related to us?
And that might be better for tomorrow, actually. So if you're able to attend tomorrow's lecture, that'd be fantastic to ask for them, because that's exactly how individuals are looking to develop the vaccine, is through understanding how the antibodies are generated, isolating the antibodies if possible to understand and characterize them, analyze them, and so on. So that may be better for tomorrow's session.
So is that Dan and Stephen, because their lecture's on Thursday? Yeah. Tomorrow.
Oh, sorry. My sincere apologies.
That's okay. Tomorrow's recycling plastics with Professor John Ward, which is a really interesting one. Yes. Dan and Stephen will be doing vaccine manufacture on Thursday, and there are still places available for that, although it's been a really huge response. So we've got 5 minutes left. So somebody's got some technical questions. Can you see the questions, Qasim?
I can indeed. So I might just kind of pick a couple.
Okay, whilst you look through those, what I'll do is I'll just quickly mention that we've got events. We've got another lecture tomorrow, which is on recycling plastics using synthetic biology from Professor John Ward, and then we've got manufacturing vaccines on Thursday. So if you've got some of these questions, might be really good to address to Professor Dan Bracewell and Stephen Morris. And we've got some questions that are coming in about admissions. So what I've done is I've put some links to our admissions pages, because if you've got questions about what entry requirements there are. Somebody's asked about how much physics is involved in the course. What I'm going to do as well is I'm going to schedule some events, some general admissions events over the summer, when you can talk to our admissions tutor, and also you can talk to some of our teaching team, and you can talk to our students as well. So it'll be an opportunity to ask them questions. So if you want to know about, how you want to prepare for coming to university, what things you might want to read, I'd strongly recommend keeping an eye out for emails coming through from UCL centrally. Something like biochemistry and biochemical engineering, this is something which we can address in a sort of biochemical engineering undergraduate degree talk, and we can go into much more detail, and you can interact with people as well.
Yeah, absolutely.
So,
what I might do with the remaining few minutes that we have is try and answer some of the questions that have been raised. So I'm going to try and pick different questions, some a bit more technical, some a bit more general, and not about the wider landscape. So there's one question here: how does CAR T therapy differ from CRISPR? So CRISPR is a form of gene editing. So it's a tool that we can use to edit the gene and actually make precision edits, so to speak. And there's other tools as well called TALENs and ZFM zinc finger nucleases. These are different tools that we can use that allow us to make specific changes or edits to a particular genome. So that's will be very useful for gene therapies. So in the case of CAR T, how does CAR T differ from CRISPR? The way to think of it is CRISPR is a tool that we can use to develop or improve CAR T therapies. One example is, so that video you saw of Emily Whitehead was an example of a CAR T therapy and the CAR T therapy process. And you'll notice they took Emily cells and they engineered them in the lab and then gave them back to Emily. That's what we call an autologous process. It can only go back to Emily. It's patient specific. It's Emily cells, has to go back to Emily. But there's a lot more value if we can develop what we call allogeneic processes. So let's say we find a super donor. I don't know, let's say for argument's sake, we've got Kim, who's a fantastic individual. His cells are amazing. Kim is happy to agree to donate some of his cells. He's perfectly healthy, happy to donate some of his cells. We can then use those cells using CRISPR. We can then, if we were to give Kim cells to someone else without editing them at all, if we put the CAR gene in but didn't edit them at all, it would cause an immune reaction in the people that received it. So the patients who received, these cells would have an immune reaction, and it would be very unsafe. So, but what we can do is, using tools like CRISPR, we can remove or knock out the genes that cause the immune reaction and cause the body to realize, hold on a minute, these aren't my cells, these are someone else's cells. We can remove those genes, but still have the CAR gene in there. And suddenly now we've got an allogeneic therapy using CRISPR. So that's one example of how we can use CRISPR to significant, and that is actively. So one of my fantastic postdoctoral researchers, amazing individual, is now gone on to work for a fantastic company in France called Selectis, who are the first company to look at allogeneic CAR T therapies. There's one or two final questions, which I think are relatively short, and then we'll close here. So apologies if I didn't get to answer any of your questions. Do feel free to email me afterwards. I'd be more than happy to discuss as well, or come to our open day. There's a question here which is slightly different. I have a question related to investment. Do you think cell therapy will replace antibody drugs to become the next investment hot trend? So they won't replace antibody drugs. Antibodies, I think, of the top 10 at the moment best-selling drugs in the world, 7 are antibodies. They're not going to replace antibodies just yet, but they will in the future, and they are already. So in terms of investment, a lot of the investment in the biotechnology sector is going into cell and gene therapies. So GSK are plowing huge amounts of money, and all of the major pharmaceutical companies are, and all those spin-out companies you saw from UCL are all cell and gene therapy focused. They've all received, so 2 or 3 of them have had initial public offerings, so IPOs, and gone to stock market with over $100 or $150 million generated. So that's phenomenal. So there is a huge amount now that's emerging, and that's something we go into in the master's program. The final, I think we've got 1 minute left. I'm pushing Kim here. Sorry, Kim. The final the question that I'll probably answer is, does UCL have the facility to research the manufacturing of cell or gene therapy at the industrial scale? Absolutely. So for the industrial scale, depends on what we're talking about. If we're talking about autologous, which is patient specific, we only need maybe half a litre or a litre. And we've got systems that are currently being used to manufacture these therapies for patient specific therapies at the moment in our labs. And we're doing a lot of the research on that. For allogeneic, we can go up to 50 litres, which is actually, for cell and gene therapies, is probably maybe all we need. But if we were to go higher, we often collaborate, and this is one of our main industrial collaborations with various companies, is looking at the scalability and addressing the issues of scale. It's not as simple as saying, well, we're going to go take a small scale and just put it into a larger reactor. Doesn't work and never works like that. There's always challenges, and that's a lot of the focus of our research. I now notice it's 2 o'clock. I don't want to keep you away from the other activities you have planned for today, but it's been an absolute pleasure. Thank you all for joining this particular lecture. Do join tomorrow's lecture on recycling and plastics and the biochemical engineering input into that, and also the lecture on Thursday around vaccine production, which it's getting a lot of attention. And I like to joke with my colleagues, vaccines are great, and clearly we absolutely need vaccines, but I think cell and gene therapies are the best, personally. And with that, I'll finish.
Great. Thank you very much, Qasim. Thank you, Rana. I'm really grateful to everyone for joining us and for these excellent questions. I've put some links up to our news and events page. You can see links to tomorrow's lecture on recycling plastics with synthetic biology, the vaccine manufacture event with Dan and Stephen on Thursday, and of course Qasim's MSc Virtual Open Day, which is taking place next week. All three are places available, and what we'll try and do is Qasim and I will work offline to answer these questions so that there are some kind of responses there for you as well. We'll also edit a recording of this for the lecture itself, and a link to the video that Qasim showed, because I've seen it many times, but every time I see it, I still find it very moving and very interesting. So thank you very much for joining, and we will be in touch with further details. Okay.
Thank you very much.
Goodbye.